Thank you, Larry. Sorry about, about the visual complication. So it is my pleasure today to introduce Professor Larry Ziegler. He's an old friend. I don't know how long ago he gave an MOS seminar, maybe 6 or 7 years ago. And we figured that it's a good time to invite him over here again about telling us some of his new work on doing ultra-fast spectroscopy. I guess IR is one area, but looks like today we are more on Raman. And I think that we would have a treat about looking at how to use ultra-fast spectroscopy for many applications, including metabolics and forensics. Thank you very much, Larry.
Okay, thanks, Peter. So I'm not going to talk about ultra-fast work today. I was going to tell you about our adventures over the past, now it's been 13 years or so, trying to exploit the phenomenon of surface-enhanced Raman spectroscopy for a variety of biomedical applications. And so here you see the title of my presentation. I probably don't have to convince this audience that there are certain advantages of using spontaneous Raman spectroscopy for biomedical applications, but I will highlight what I see as advantages. And that is certainly molecules provide the inherent labels themselves. You don't have to provide any additional extrinsic labels or reagents. The other attribute is I'll say it this way, the free induction decay times of the vibrational resonances. They're relatively long, which is to say that you have narrow features that describe a vibrational spectrum, so that vibrational features then are present for a given species, and you have a number of these. So there are a number of features that identify a particular molecule. And then because they are so narrow, it's inherently a multiplexing technique. You can see the multiple components that may be present in a biological fluid. Of course, the sample size can be quite small. Right? Just the diffraction limit is, at least in spontaneous Raman, is the size that we can minimize this technique for. So that gets us down to single cell kinds of investigations. It's fast, easy to use, certainly made portable. It's non-destructive, which for some applications can be valuable. In principle, once you interrogate the material by light, it's not resonant, and you can interrogate that material by other techniques. And it has certain advantages relative to IR, in particular the diffraction limit, wavelength size, and also some limitations of water. Water is much more of a problem for the infrared than for Raman spectroscopy of biological fluid. There are, of course, some disadvantages with spontaneous Raman. It's inherently a weak effect because it's a two-photon phenomenon, light in, light out. And furthermore, fluorescence can be a problem. So surface-enhanced Raman spectroscopy, which I suspect everyone here has heard of, has, of course, all the advantages that I just described for spontaneous Raman spectroscopy. But in addition, it offers high sensitivity that allows you to look at solutions or biological fluids and test for or analyze for low analyte concentrations. Molecular enhancements in soluble average can be 10 to the 6, 10 to the 8, something of that order. And that's key to some of the applications that we've been able to find valuable to exploit service for, such as growth-free bacterial diagnostics. The other attribute, which is quite convenient for service, is that because of efficient energy transfer, there's a fluorescence control. So fluorescence is never an issue, as it might be for normal spontaneous Raman. So again, I presume everyone here has heard of surface-enhanced Raman spectroscopy. It's been around for 40 years or so. I did say already the per molecule enhancements are 10 to the 6, 10 to the 8. So, appreciate that means that the local electric fields, as a result of proximity to a nanostructured metal surface, due to the surface plasmons, are enhanced, right? The field itself is enhanced by maybe a factor of 50 or 100. That's the physics that we're going to exploit to make this bioanalytical technique have value. Of course, once again, due to the enhancement mechanism of surface-enhanced Raman spectroscopy, it's only those molecules that are within a couple of nanometers of the nanostructure of the metal surface, which by the way is typically gold or silver, that will be enhanced by this phenomenon. So some of the SIRS-based biomedical applications that we've explored and received support for and some strides with are listed here, and I'll touch on some of these. Blood infections, that's really hard. In fact, I'm not going to be talking about that. It's bacterial, urinary tract infections, sexually transmitted diseases. And then you've seen interesting applications in blood diagnostics, learning about aging, and then I'll show you something about the rapid malaria detection methodology, cancer cell identification, detection, and then most recently we received some support to apply this technology in the field of forensic science. And aside from these very applied kinds of applications, I think also serves as the capacity to provide new information on in vitro biological processes, in particular, perhaps be exploited as a novel tool for systems biology investigations, which hopefully I'll be able to convince you of in a couple of minutes. So the platform that I see for these biological applications that are based on SIRS, frankly, have four key components that I can identify. One that's crucial to get this work started is sample preparation that generally enrich, in the case of bacterial diagnostics, enrich bacteria from whatever biological fluid they're in. This step, interestingly, can often be the slowest step in a methodology that I'm calling fast. The second component is the SIRS substrate itself. And I'll show you a little bit more about that, but just briefly, it's a gold or silver nanoparticle covered SiO2 substrate. And again, I'll say a little bit more about that. A SIRS acquisition instrument is also required. I've built several prototype portable Raman microscopes. Most recently built with a company called BioTools. And then software for detection and identification. And again, we've developed some novel multivariate data analysis approaches for enhancing identification of a bacterium spectrum within a library. And I'll just touch on that in a moment. So just a word about the SIRS substrate, because that really is the magic bullet, so to speak, that makes all these applications go. What we found most effective is a substrate we developed some time ago now. It's it can be gold or silver, but most of the work was done with gold. It's produced by a metal ion doped sol gel procedure. You end up with a piece of SiO2 that is covered, physiabsorbed with clusters of gold nanoparticles, typically 80 nanometers. Here you see a sense of the surface roughness of these gold particles relative to a two-cell organism, Bacillus anthracis sternus. It's a non-virulent Bacillus anthracis. Anyway, just give you some idea of the sense of roughness. The enhancement factor, per molecule enhancement factor, the sample average is between 10 to the 7, 10 to the 8th. And the shelf life for these gold substrates, we've gotten to be as long as about 5 months. It costs nothing to make because they're made by the chemical process. I've got no religion about the SIRS substrates. I'm just interested in whatever works the best. And we thoroughly searched a variety of engineered substrates. So I'm just listing some of the candidates we interviewed, so to speak. And our chemically produced sol gel substrates are the ones that end up working the best in terms of signal to noise. So that's mostly what I'm telling you. So just give you some overview here. These are SIRS spectra of, I guess, 8 different bacteria covering 5 different genera. They're both signal to noise, a couple of milliwatts of incident power. Everything I tell you also is at 785 nanometers. So purely non-resonant with any features in the bacteria. And I think already by eye you can see that these spectra are different for these different species. Although you can also see that there are a number of features that they appear to have in common. And so, to me, a very interesting aspect of this work has been trying to understand what the nature of this signal is, and I'll come to that shortly. Okay, this just gives you some overview of our capabilities. I did want to just mention this one multivariate data analysis approach that we use, this little trick that works great. And it's completely counterintuitive for anyone who's a spectroscopist. And that is demonstrated here by this little example. So here I have Certa spectra of 4 different closely related bacillus bacteria. And they're actually a subgroup of Bacillus cereus. So they're very closely related. And what I do is a principal component analysis, which is that's simple. I mean, that's well known. Just press a button for that in any kind of package. But the trick is that our input vectors are what I call barcodes. That's to say, I take the sign of the second derivative, just curvature up, curvature down, as a function of frequency. Convert it literally into a barcode, zeros and ones. And then I use that as my input vectors. And well, I had no expectation this would work great, but it really does. And here I'm showing you a principal cluster analysis plot, just a 2D plot. But just do the normal thing. This is the cluster picture I get by just taking the normal thing, just taking input vectors of spectra. The tightness of these clusters tells you something about the reproducibility of the vectors are. And of course, the distance between the centers of these clusters tell you about specificity. And so you can see how this would be used potentially as a diagnostic, and I'll give you some examples later on. But the point is, if I use the input of these barcodes, so I throw away intensity information, right? So we don't do that often in spectroscopy. Then you can see the same data. Here's the tightness of the clusters, and of course the distance between the clusters is pretty intense. So you increase the sense of the reproducibility and also the specificity. So that's it. That's just one little advertising about this counterintuitive trick that works so nicely. So let me give you an example of how this might work for a rapid antibiotic-specific bacterial diagnostics. And the example I have in mind is urinary tract infections. These are amongst the most common type of infections on the planet. More than half of women on the planet will have a UTI, and a significant percentage, something like 20% of those people will have chronic infections. So this is a big problem, common problem. E. coli strains are the major cause of UTIs, between 50 and 70%. And then there are just a handful of other species that contribute to UTI infections. A concentration of 10 to the 5 bacteria per ml in urine is the minimum standard for the diagnosis of a urinary tract infection. It's at that level that a physician would prescribe antibiotics. Current gold standard is still growth, quantitative urine culture growth. It takes 24 hours. And then to determine antibiotic specificity, which you do, of course, you grow it in different antibiotics as it grows. That's what gets prescribed. And that takes another 24 to 48 hours. So as a result of this growth period, of course, physicians typically prescribe broad spectrum antibiotics just on the basis of presentation. And that, of course, contributes to infective treatments, chronic infection, increased health costs, and probably most insidiously, the increased resistance to antibiotics. And so what you'd like to be able to do is, of course, to have a rapid, and time scale to me that makes most sense at the time that the person would present themselves to a clinic before they leave. So let's say an hour. That's a time to get information so that you can have an antibiotic-specific, low cost, easy to use methodology for UTI diagnostics. Okay. So here's some of our attempts to develop a procedure this way. We can spike urine with bacteria. So I haven't done anything with clinical samples. We just spike urine with bacteria in there for a while. And then we filter and centrifuge, so nothing fancy. This can really all be nicely done in a microfluidic fashion. The enrichment that results from the centrifugation filtration is about 3 or 4 orders of magnitude. Take 1 microliter of that enriched material, put it on a sort of substrate, and within a couple minutes you get these representative sort of spectra. Okay, these are 3 different species. And I will make the point once again that there are features that they do have in common, and certainly features that are distinct. And that if I just turn my spectrum into these barcodes that I told you about, and put this in a PCA treatment, you can nicely separate them. Okay, so you get the idea how this can be used potentially as a diagnostic. Let me give you a further example. Oh, we've got sensitivity burn. This is now down in order of magnitude 10 to the 4, so that's an order of magnitude lower than a physician would need for making a prescription, or write a prescription for antibiotics. So we have lots of sensitivity there. So here we spiked urine at the 10 to the 5 level, that minimum clinical level, with 4 strains of E. coli, 1 strain of Staphylococcus and 1 of mirabilis. And then leave them for a while, pull them out, take this sort of spectra, and the point I want to much appreciate in order to see how this would have value is that our library is already informed by the drug susceptibilities of each of these strains. So it's not just species, it's strains, right? So I have information on the drug resistance and susceptibility. So we came back weeks later, grew up one of those strains, and sure enough, it clustered with this 11996. So within an hour, for example, this was carried out by physicians. The physician would know, that's this guy here, would know that the drug to offer up was amoxicillin, for example, and not Cipro, which is the antibiotic that is often prescribed for UTI diagnosis. So that's the idea. That's the spirit of this. And of course, now the idea is to extend it to a larger number of strains. This is a large number. And these are clinical isolates that we gave us, and we've got their predetermined drug susceptibility profile. And now we use a slightly more sophisticated multivariate data analysis approach. It's still barcode-based, but it's partial least-squares discriminant analysis, getting more degrees of freedom that we have to keep track of. And with this classification model, we achieve 100% sensitivity, 96.4 specificity. These are analytical, true positive, true negative measures. So it seems to work pretty well, even given the fact that we're looking at different strains as well as species. And then, the goal here is to push us on to larger data sets and develop other strategies to handle identification, such as, which is what we're doing now, combining a hierarchical cluster analysis as well as a PLS-DA approach. Okay? The STDs. So let me tell you how search can be used for STD diagnostics, in particular, distinguishing chlamydia and gonorrhea. So these are both diseases that are caused by gram-negative bacteria. Chlamydia is caused by Chlamydia trachomatis. Gonorrhea is an infection due to Neisseria gonorrhea. I don't need to convince you that effects of untreated disease can be very dangerous. Again, classic STD detection is cell growth, and it's very slow. These are organisms, quite frankly, that grow much more slowly than the other bacteria that I spoke about. Very successful, where how they found a place to live on the planet, but very, very slow. The That's current STD diagnostics are these nucleic acid amplification as they're quite sensitive, but at the same side, the cost of sensitivity is contamination, and that does tend to be a problem. They're relatively expensive and not done routinely. So I looked, I went online and, well, if you had a concern, you can, in 1 to 2 days, get what's called the fastest results possible by this company in Boston for $189. The other downside from this is to determine whether the bacteria that are causing infection are living or not. You're just testing for the presence of nucleic acids. And so it's not a tool that can be used to determine whether the treatment is effective. Okay, so again, you know, one certainly potentially could have an impact here with the kind of methodology that we're trying to exploit. Chlamydia is treated with one type of antibiotic, gonorrhea with another, and they often co-present. 40% of the times they co-present. So someone shows up with a problem, and generally both drugs are prescribed. And of course, again, leading to this insidious problem of overprescription of antibiotics. So certainly have a detection identification procedure that was limited by growth kinetics, means that you would diminish progression of disease, disrupting transmission, this is euphemism, and reduce, of course, the overprescription of antibiotics. I just mentioned all things that I learned and looking for different ways to exploit this technique. The life cycle of chlamydia is a bit unique in that it invades host cells as these reticulated bodies, and then it lives on the nutrients inside of the cells. And then after several days, they explode these extruded bodies, and the whole process starts all over. So I'm pointing it out for a reason. You'll see it for a couple of months. So they are a little different. So fine, it's just my candy at this point. I'm showing you on the right hand the Search spectra of chlamydia, both on gold and on silver. And on the left here, this is the causal agent for gonorrhea, right? Both on gold and silver. So a couple points to make here. One is that clearly each of these organisms has a unique signature. That's so you know one could identify one or the other, and in a mixture they should be easily distinguishable, right? They look very different. The other intriguing thing is the difference between gold and silver. Not so much for the gonorrhea, that's fairly similar. The chlamydia, of course, looks remarkably different, whether it's on the gold or the silver. I'll come back to that, but the main point is, you just at this point, just a pure application of phenomenology. Yeah, this really does look like a methodology that would be able to distinguish these bacteria and potentially useful for rapid identification. I think it's really crucial to get beyond just the phenomenology and really understand what the molecular components are that are contributing to these signatures. Right? It's important for lots of reasons. One, so that you can develop a robust, reproducible signature in itself, and develop the best methodologies and the things you can do, which figure out where the molecular origins are, maybe things you can do to enhance those signals, and develop strategies for enriching the materials that give rise to these signatures. I think for adoption in the medical community, it's important to be able to offer an explanation that is not just phenomenology. Then, of course, there's the potential for exploiting the signatures that you see for learning about cellular biochemical activity. And of course, I'm continuing ahead that it could have a role for systems biology investigation. And then, finally, to broaden this technique for other applications, which understand what the signatures are used for. Let me first make the point that the Search spectra excited at 785 nanometers, and that's all I've been talking about. At 785 nanometers, the Search spectrum has no correspondence to the normal Raman spectrum. So So you can acquire a normal spontaneous Raman spectrum at 75 nanometers, as I'm showing here. You wait longer, you have higher concentration, and you can acquire a Raman spectrum. And so I'm just comparing here for two different species, for E. coli and some salmonella, that the SIR spectrum, which is in blue, is completely unrelated to the normal Raman spectrum. And that's just as it should be, right? If I'm acquiring a spontaneous Raman spectrum, then it's, of course, the number density weighted molecular Raman polarizabilities that are giving rise to these sort of signals, and that's going to be dominated by the cytoplasm, right? That's where most of the volume in the cell is. On the other hand, these are the distance dependent of the surface mechanisms. All those molecules that are close to the surface, whatever they might be, that are going to give rise to these sort of spectra. So, of course, you'd expect them to be quite different as they are. So what do they do to it? First of all, let me jump to the conclusion and then show you all the evidence that justifies this. The SIR spectra of bacteria, at least most of them, are dominated by metabolites. So how do I know this? Well, our first clue is simply just looking at the SIR spectrum of bacteria and the number of purines. So for example, the black line here is the SIR spectra of Staphylococcus aureus and strain, and the red line is the SIR spectrum of substrates and adenine. Very close. Very close. Then this is an Enterococcus faecalis, black. This is spectrum of hypoxanthine, this guy here, very close. And likewise for this Agalactia mamani, you see a number of peaks in common between the bacterial SIRS signature and that of xanthine. Okay, so that was my first clue. As any good vibrational spectroscopist, you want to figure out what you got. Well, good old-fashioned isotopic substitution. So I can bring that to the table. So here, this is again, that's Staphylococcus aureus. I've grown it in nitrogen 15. And so sure enough, you see shifts, as you might expect. And just comparing it with adenine, and the shift is virtually identical for nitrogen 15 adenine and compared to nitrogen 14 and definitely organism itself. The Anthracis stern is a little more complicated. That is contributions from adenine, hypoxanthine, and guanine, which I'll tell you about in a moment. But the point is, when I put the nitrogen 15 version of those molecules in, I get the same shifts. So, you know, now I really feel like I know what's going on. But of course, we're going to finish. Where we grow these bugs, and we say, you know, we capture them whether they're growing in blood, urine, media, and then we wash them in water. Bacteria are very hardy, so you don't have to put them in a buffer. Just still water, and they're fine. If distilled water was a problem for bacteria, you know, we'd have a very ubiquitous antibiotic. So they're very hardy. We took the water that they're sitting in after we washed them, supernatant, and we enriched it with the lyophilizing, and then put that on our sort of substrate. So no cells, just the enriched supernatant. And the SIR spectrum of the enriched supernatant was virtually identical to that when we just put the cell. So, you know, that's telling you that the SIR signatures come from small molecules, these concentrations of micromolar or so. They're water soluble, small molecules, and giving rise to these signatures, not the cell wall. And that's frankly what I had told people for many years about this work, that there's cell walls, unique cell walls that give the signatures, and that's what's mostly in the literature. That's wrong. The final bit I do want to show is of course, now that we have the supernatant, then we can use mass spec to just verify what we see in the SIR spectrum. So here for Staphylococcus aureus, which I told you is dominated by adenine. This is a mass spec spectrum. The tall trees here have nothing to do with adenine. The
acid, aspartic acid. If I go up